Hi, I'm Karen Green. I'm the product manager at Ecolab. I oversee their chromatography portfolio, and today I'm going to talk about how pH can impact the elution of especially some complicated molecules. So monoclonals are still the dominant antibody that is used for therapies in the drug industry, but however, we're finding other molecules like bispecifics, multispecifics that are coming to the market, proving their efficacy in treating diseases. But what's happening though is they're very complicated to purify. So the traditional protein A's that we normally use aren't giving the purities that are required. They're not giving the elution pools are larger because you have to elute longer. And these are problems to the industry. So what the Jetted A50 HipH has brought to the market is a specific tool to be able to deal with these types of molecules. And what it does, what it allows, is because it's specific for the higher pH does allow for that elution to happen with higher yields and higher capability. And then at the end of the day, with a smaller pool of elution, which does help customers further on down the process of the bioprocessing scheme. So as when molecules are brought to market, they have to be cloned, and then multiple runs are done to get that specific molecule that's going to be the best candidate. And during that process, because of being able to, again, elute with the high pH, we're able to get those higher yields, that higher purity. There's a split of the molecule itself and the aggregates that can come along with the process. And because it's so pure, it's able to, there aren't any other additives or anything, and it's actually, it can go right on down to the discovery mode, from discovery to development, and be able to move on down the line. So customers have to be able to provide viral safety clearance data when they submit for their drug molecules. There are requirements that they have to meet certain log reductions to get those FDA approvals or those global approvals for that drug to be marketed across the globe. So what is necessary? Chromatography can be used as a viral clearance step, also filtration and chemical. But during that step, the normal protein A does have a viral clearance of 2 log reduction. What we're seeing with the Jetted A50 HipH is a 5 log reduction, and that can contribute to the overall viral clearance efficacy of the product, and that's a good thing. As I mentioned earlier, molecules are starting to become more complex. It's not just the monoclonal anymore. There's bispecifics, there's multispecifics, there's FC containing molecules. They need different types of application strategies to be able to purify them to the levels that they need to be purified at. So for example, the high pH does meet the need for those types of molecules. There are also molecules that contain the CH1 human domain. CH170 is a product that we have that will provide purities and clearances that customers need in that capacity. We also, if a customer is looking for something that needs multiple cycles and has high resistance to caustics, which is one of the steps within the process of purifying antibodies. So the Jetted A50 technology does allow, it's a uniform bead, it's across our whole portfolio. What it brings to the table for chromatography is high mass transfer, which is good for that high purity, and then also very good flow rates, which also decrease the amount of caustics that are required when running the processes. Also very close to the heart of Ecolab is around sustainability and reducing caustics into the footprint of the environment. So the whole Jetted A concept is something that is very relevant and creates sustainability moving forward with the chromatography process.